The plant cuticle is a waxy outer covering on plants that has a primary role in water conservation, but is also an important barrier against the entry of pathogenic microorganisms. The cuticle is made up of a tough cross-linked polymer called cutin and a protective wax layer that seals the plant's surface. The waxy layer of the cuticle is obvious on many plants, appearing as a shiny film on the ivy leaf or as a dusty outer covering on the surface of a grape or cabbage leaf, thanks to light-scattering crystals present in the wax. Because the cuticle is an essential adaptation of plants to a terrestrial environment, understanding the genes involved in plant cuticle formation has applications in both agriculture and forestry. Today we'll show the analysis of plant cuticle mutants identified by forward and reverse genetics approaches.
Hi, my name is Patricia from the laboratory of Ljerka Kunst at the Department of Botany at UBC. Today, myself and others are going to show you how these plants, Arabidopsis, can be studied to understand how plants make cuticular wax.
I'm Miao from Reinhard lab at UBC. I'll be showing you how we analyze chemical composition of cuticular wax using gas chromatography.
And I'm Allan DeBono from the laboratory of Lacey, also at the UBC, and I'll be showing you how we analyze the crystal structure on the surface of Arabidopsis using cryo-scanning electron microscopy.
Our overall goal is to understand how plants take ordinary fatty acids of 16 or 18 carbons, extend them to very long chains of 26 to 34 carbons, then modify them into protective lipids that we find at the plant surface. So let's get started, and I'll show you how we identify cuticular wax mutants using forward and reverse genetic approaches.